Hello, and welcome to part 2 of chapter 24. Where we left off last time, we were talking about the overall filtration process. And so now we're going to make a little discussion about the filtration membrane. So remember, filtration membranes in the glomerulus, visceral layer of the capsule. All right, so that the capsule is what engulfs the glomerulus, remember. So the visceral layer is what would touch on the glomerulus. Okay, it is actually the podocytes of the glomerulus. So it ends up being the endothelium of the glomerulus, the outside of the glomerular capillary. Now remember, this is a specialized kind of a capillary, and it is fenestrated. So fenestrated just means it has holes in it. So the basement membrane has holes in it, and what it has on those in that layer are podocytes, like I was saying before. Podocytes like podo, like being feet. Okay, pedicels, long process pedicels, pedi like pedestrian, right, walking, so feet. And there's slits in between those. Another type of cell that you find in the visceral layer is mesangial cells. And we'll look at for a minute how they work as well. So here is inside the corpuscle. The juxtaglomerular apparatus is right here. Again, controlling the afferent and efferent arterioles and regulating flow and doing other things too. Excuse me. You can see where the mesangial cells are in between the podocytes. So these big cells with the long projections from it are the podocytes. Looking even closer, you can see how the podocytes overlap each other, leaving this little slit in between them. And it's through these slits that cover the holes, the fenestrations, that the filtration occurs. So the fluid comes out there. I'm going to get even closer here. And you can see here's the pedicels, okay, right here, and how they cover up the fenestrations. Maybe some of them completely, some of them slightly, right here, letting through just certain amounts of substances. So it's very limited in what it can, it's variable in what it can allow through, okay, according to conditions. So how does the filtrate form? Well, it comes out of these little holes in the fenestrations and gets regulated by the pedicels of the podocytes. The mesangial cells also play a role in that as well. So with three things in the blood, okay, freely relative to what gets filtered, the filtrate. Freely filtered, pass easily through filtration membrane, like it says, small substances, water, glucose, amino acids, whatnot. And then that which is freely filtered, that which is not filtered, and that which is filtered on a limited basis. Okay. What do the mesangial cells do? They phagocytize material trapped within the basement membrane. So that's another thing that they do. So we're looking at pressures here. So the hydrostatic pressure, again, is just like the pressure that we looked at in the circulatory capillary, all right, where it is brought to the capillary by the pressure that was given it by the beat of the heart. But in this case, it's a little bit different, and we'll talk about that in a little bit, because the afferent and efferent arterioles control the pressure in there by controlling the flow in and out of the glomerulus. All right, so the hydrostatic pressure in the glomerulus is going to want to push the fluid up out of the through the holes and out of the blood flow. What opposes this? Blood colloid osmotic pressure. Again, something that we looked at before in a regular capillary. So remember now, what comes out is more dilute than what's left behind, right? Because there's smaller substances in it. So because it's more dilute, there's going to be an osmotic pressure for the fluid to want to go back in, okay? This is colloid osmotic pressure. And then there's a hydrostatic pressure within the capsule itself. All right, so what we were looking at here, inside the capsule itself here, there's a hydrostatic pressure. So these are opposing that filtration pressure to push out the hydrostatic pressure inside the glomerular capillaries to push fluid out. But it's still, even together, they're not enough to overcome the pressure. And so there's a 10 millimeters mercury pressure that pushes fluid out of the capillary, the glomerulus. Okay, so there you go, right there. You can see how they all add up together. Now, this pressure is what influences the glomerular filtration rate. So there's the rate. So the rate at which volume filtrate is formed. So what volume comes out of that the blood and creates a filtrate? What volume does per per unit of time, 1 minute usually. And this can be altered, can be varied. The glomerular filtration rate can be varied according to